Well, I'd like to thank the organizing committee for inviting me to be a part of this symposium. It's always been an honor to me to be associated with Professor Fano and his students and colleagues. It's also been a great opportunity for me to get up to date on the many areas of atomic physics, still feeling the influence of Fano's work. And again, it's also an opportunity to address Fano's students and colleagues as a group and bring to their collective attention a fundamental inequality that I think this community as a whole is unaware of. I've coined a term for this equation. Called it the Baer inequality, and fundamentally it appears like this. This may appear to be a contradiction and absolutely impossible, but as with most analytical expressions, if you express it graphically, it becomes much more clear. The Tom Baer on the left is actually named Tom Baer, and he's a distinguished professor at the University of North Carolina, and has spent the last 25 years doing experiments in photoionization and molecular dissociation. The Tom Baer on the right, besides being a Brazilian soccer fan, is CEO of a biotechnology company in Mountain View. And Fano students always get these two people confused. In fact, it happened just before this talk. So I wanted to set the record straight. The organizing committee, being composed almost entirely of Fano students, was unaware of this inequality, and they invited this Tom Baer to give a talk at this symposium. So rather than hearing a talk on Fano resonances and photoionization, you're now going to hear a talk about quantitative molecular cell biology. In defense of the organizing committee, I do have a PhD in atomic physics from the University of Chicago, and I did actually work with Ugo Fano. I think by any measure I was a student of his, at least for a year. He paid my salary. He hired me in order to explain an experiment I was doing with Isaac Abella. We were looking at photon echoes in cesium gas in a magnetic field, and Fano did not understand the results we were seeing. So I worked with him and developed a spherical tensor description of that whole experiment, and it became part of my thesis. Part of the challenge in working with Ugo is that he was so far advanced in his field that you really had to learn to speak physics in his language and had to learn how to think physics in his language. And this is one of 2 times in my career that I've had to do that. It was well worth the effort. It took about 6 months to be able to understand what was being said at the lunchtime conversations in his group, and I just sat patiently listening until finally I could begin to understand the approach to physics that really characterized Fano and his students. The second time this happened to me was also an NIST alum, Jan Hall at Gilla, and when I went there, I also, for the first 6 months, could not understand what he was saying. Ended up spending a 2.5-year postdoc with him, and once again, it was, at least at that, I knew what I had to do. I had to learn another language to be able to understand it. In both cases, it was well worth the time spent. Fano influenced my growth as a scientist, I'd say more than any other person I've worked with. He instilled in me a real love of theoretical physics and applied mathematics, and that's really stayed with me throughout my career. So what I'm going to be talking about today is work that I've been doing at my company, Arcturus. It's a company I started about 6 years ago. To back up a little bit, about 10 years ago, I decided to change fields. I moved into the area of biotechnology and biomedicine, really because I found the science to be absolutely fascinating, and I knew nothing about it, and I wanted to learn about it. So I stepped down from position at Spectrophysics, where I was VP of R&D, and joined a biomedical company developing instrumentation for AIDS diagnosis and monitoring, and headed up a multidisciplinary research group there and just began to learn a whole new discipline. What's really interesting about the revolution that's occurring in biotechnology is that it really is a technology-driven revolution. There's no fundamental new paradigm shift that's happening. It's just we have the technology now to measure what's going on chemically with a precision and a specificity that we've never had before, and a sensitivity we've never had before. This has been evidenced by the development of gene sequencing technology that occurred in the early 70s and was automated in the 80s and 90s, which led to the Human Genome Project, which has been going on for the past decade. And that has led to the sequencing of the human genome. We now know about 2/3 of the genes that are active within the human body as a result of this project. It's been a massive effort. What's happening now is that this database is being used to actually understand where genes are active within different portions of the body, and it's leading to a whole nother revolution that I call basically functional genomics. It's these genes and this data that's been discovered as part of the Human Genome Project is now this data is being applied to understand basic cell processes, the differentiation between normal and disease cells, among other topics of study. Similarly, it's being driven by technological innovation, and I call this new technology microgenomics, and that's what my company Arcturus is involved with, developing technology for understanding gene function within the human body. This is leading to a revolution in our understanding of molecular cell biology, and that's what I'll be talking about today. My company is located in Mountain View, California. I started the company about 6 years ago. We have about 105 employees. A definition of microgenomics that I use is the quantitative molecular analysis of single cells or small groups of cells representing pure clonal populations of cells characterizing their in vivo state. And you'll understand why this is of importance as the talk proceeds. Basically, genes dictate physiology and disease in the human body. The human body is a complex organism, but it can be broken down into basically roughly 10 different organ systems within the human body. These genes are the fundamental basis of how we go about growing and developing. And there are about 40,000 genes, estimated to be 40,000 genes in the human genome. These genes encode the production of about 250,000 different proteins, which then give rise to roughly 400 different cell types in the human body. The cell types, I believe, are similar to atoms. They are a fundamental quantum of life. They're the smallest discrete living portion of the human body. The cell types can be defined by differential gene expression, which genes are being activated to make the proteins. The proteins that are produced by the cells really differentiate the cell types. And normal and disease cells can be distinguished by different gene expression. I'll be showing you data to support that. These cell types, 400 cell types, combine to form 30 different types of tissue, which then go into these 10 systems which form the human organism. So this is a sort of level of complexity. Really, in my view, the cells are the key. And maybe this is because I'm an atomic physicist and I like small round things. But really, the cells are the key to understanding, I think, the basic organism. And I correspond the elements of the periodic table, really, to these 400 different cell types. And the gene expression data that we're collecting is very similar to the type of spectra that we have been working on for the last 100 to 150 years that distinguish the different elements of the periodic table. Let me show you what are some of the challenges in doing microgenomics. Here is a typical series of biopsy slides that show cancer progression. And this is the typical biopsy cross-section. This distance scale here is roughly about 300 microns, to give you an idea. Each one of these small dots here is a nucleus. It's about 5 microns across. This is a very complex piece of tissue, but this is typical for any biopsy. It's about a tissue slice that gives you a single cell layer, so it's about 5 to 10 microns thick. Consists of a half a dozen to a dozen different cell types. This is the terminal lobe of a female breast cancer patient, and this is a normal milk duct. And these are the cells we're interested in right here. These are the epithelial cells that actually produce the milk proteins. And what we're interested in doing is studying the genes that are up or down regulated within these healthy cells, but they're lost in this mixture of heterogeneous tissue. And what we want to do really is be able to isolate these cells in order to measure what's going on in these healthy cells. This is early stage cancer from the same patient. These are the same epithelial cells, but now they've started to multiply. They've lost their growth control checks, and they're multiplying out of control. It's early stage cancer because it's still surrounded by what's called a basement membrane. It's an intact tissue, and this is fairly easy to treat. It's a lump, and you just pull it out. The next stage of cancer is invasive cancer, and this is where you no longer have the basement membrane, and now these same epithelial cells, the same healthy ones, have mutated one more time and are now penetrating into the surrounding tissue. Once again, we've got stromal tissue, fat tissue, some inflammatory cells in here. It's a big mixture, and what we're interested in is just these cells here that are located here, which are the original epithelial cells that have mutated and are now cancerous. The final stage of cancer usually is metastatic cancer, and these are, again, these same epithelial cells, but now they've changed. They've lost, one more time, they've lost their self-adhesion molecules, and they've migrated to the lymph node. And again, this big mess of cells here, this is lymphatic tissue and also inflammatory cells. And what we're interested in doing is basically starting with the healthy cells and going through and measuring the gene progression that takes place, the mutations that take place that get us to this stage. So the technical challenges are, first of all, obtaining a pure population of cells from the heterogeneous tissue samples, as I've just shown you, and then accurately measuring, doing analytical chemistry on these, the diverse macromolecule population in these pure microscopic samples. So it's a very difficult analytical chemistry challenge as well. There are several interesting scientific questions that come about. Along with these technical challenges. Are cells truly quanta? Are they reproducible fundamental particles defined by their gene expression patterns? Really, we don't know that. You'd think that they would be, but that's a question we need to answer. And then are these gene expression signatures we're going to be looking at to characterize cells, are they really universal, or do they vary from person to person? Are they masked by biological diversity? So these are the sorts of questions, scientific questions, that we were also trying to answer. So here's the type of tissue samples that we are dealing with. And one of the products that my company makes allows the precise separation of these different types of cells. You start by visualizing the tissue samples that you're interested in, and being able to highlight those cells of interest that you want to microdissect. And then we have a process that's called laser capture microdissection that we developed in collaboration with a group of the National Institutes of Health that allow us now to cleanly separate out, even on a single cell basis, the different cell populations we are interested in studying, so we can cleanly follow the progression, genetic changes that go from a normal to the disease states. So that's one aspect of the technology that we've been developing. And I'll show you how this works. But first of all, let me remind you what the size is. On the screen, these are 3-foot to 5-foot size samples. This is a typical microgenomic sample, which is approximately 200 cells. To give you an idea of the scale of what we're dealing with, that's the actual sample size. This is a 1 millimeter pinhead. And what we're trying to do is measure in this 200 cell sample 20 to 30,000 genes that have been up and down regulated within a sample of that size. So that's the analytical chemistry challenge that we've got. Here is a animation of the process of laser capture microdissection. We start with the tissue slice. It's about 2 square centimeters, 10 microns thick. We then apply to it a plastic piece that's got a precision polymer film fabricated on the bottom, plus some standoffs. This is a thermoplastic film which has a dye in it which can be activated by a laser. The laser activates the dye, heats it up gently, and injects it down into the tissue. You can then walk the laser beam around the tissue sample, and wherever the polymer has touched the tissue, the cells stick to the polymer. It's basically a laser-activated Scotch tape. When you remove the cap, the cells come with it. You've now isolated the cells. We have mating microfluidic devices that completely isolate the microdissected cells. And then we apply to this digestion buffers enzymes and chemicals that digest the cell surface and allow the macromolecules to come into solution where we can do analytical chemistry. So we now have an isolated section of the tissue, and we've extracted from it the DNA, RNA, and proteins from those cell samples. So we've got half the problem done. Here is a video of the actual process that I just showed you in animation. And this happens in 5 milliseconds, so it's a very high-speed video. And the laser comes in from the left here, heats the thermoplastic, and the plastic undergoes a crystal into glass transition, which has associated with it a volume change of about 20%. But the only place that's heated is where the laser beam is, so it can't expand laterally, it can only expand out, and that's why you get it shooting out across this 50 micron gap. This gap is actually about twice the thickness of the polymer film, so it actually pulls in polymers from outside the laser beam and scoots it out. It's a fascinating process, and you can actually get this thing to shoot out several hundred microns. It's a very interesting materials processing setup. This the temperature rise that's associated with this transition is only about 30 degrees centigrade, so it doesn't do anything to the cells or the proteins within the cells. Here's an example of an electron micrograph of a single cell. This is a white blood cell, a leukocyte, that's been captured on the top of the polymer. So we can go right down to single cell resolution with this technology. These are the types of instruments. We have a manual instrument and an automated instrument for doing this type of microdissection. And here's an example of a screenshot from the automated instrument. This is, again, breast cancer. This is the cancer region in here. It's been stained to highlight a particular protein that's expressed by the cancer cells. And we can use the image processing to outline the regions of the cell so that we can do automatic image processing. The operator can then pick the regions that you want to microdissect, and then the computer goes along, raster scans, and basically sticks the tape just to those regions, and you end up with a microdissection, a clean microdissection or clonal population of cells. So we have these cells. So how do we measure the mRNA library? We're really interested not in the DNA in the cells, but what the RNA. The RNA actually encodes the proteins. This is what gets copied. And the more RNA of a particular sequence you have in the cell, the more of that protein that's made. So we want to quantitatively measure the RNA library that's within the cells. So to give you an idea how we do that, this is what RNA looks like. It's composed of 4 different bases that make up the polymer. And usually you have a gene that's encoded with about 1,000 to 10,000 bases. Then all messenger RNA has a tail on it that's called a poly-A tail that consists of just a series of A's that go from 50 to 200 bases. And we use this to recognize the messenger RNA in the sample. This turns out to be only about 2% of the RNA that's in the sample. The rest of it is ribosomal RNA, which we're not interested in. Just to give you an idea, again, of the technical challenges that we're dealing with, in each cell there are about 200,000 mRNA molecules. Of this population of 200,000 mRNA molecules, there are 2,000 to 5,000 different sequences that are contained in here. A low-expressed gene will have about 1 to 5 copies per cell of that particular sequence. A high-expressed gene will have about 1,000 to 10,000 copies. Typically, we can do the measurements we're trying to do on about 100 cells. So what we're trying to do is, again, analytical chemistry, where we're trying to measure a very complex molecule, the concentration of it, down to starting at about 500 total molecules and going up to about 5 million. So we want about 3 to 4 logs of sensitivity, and we want to be able to measure very, very low concentrations of these complex molecules. And this is how we do it. I'm going to be stopping and starting this. We start by putting the sample into a purification device. This is a device that has a porous silica substrate to it. Turns out under high salt conditions, nucleic acids will bind to this porous silica substrate. All the proteins, lipids, and other junk carbohydrates don't bind to it and get washed on through. So that's the first step, is let's clean up and just get nucleic acids. Here again is that mixture of extracted molecules from the cells. And what we do is under high salt conditions, it all sticks just to the porous silica substrate. We then come in with just normal water, and that then we switch tubes, come in with water, and we just wash out the nucleic acid. So now we just have purified nucleic acid. You'll notice in here that these are the RNA molecules with their poly-A tails. What we have is then we add a particular sequence to this, which has a complementary sequence to this messenger RNA tail, and it selectively binds to the poly-A tail. So and it has associated with it a specific primer, which has a specific set of sequences, which will be important later on. Going through this, just to give you an idea of the complexity of this process. What we're trying to do here, I didn't explain in the beginning, is what we're trying to do is amplify or copy, using enzymes, the nucleic acids that were in the original sample to get them up to a point where we can actually measure them. So the first step, again, is hybridizing this primer A to the tail of the mRNA. Oops. That's all right. Let me start that over again. It'll go quick. So we hybridize to the tail of the mRNA with a particular probe sequence. And then we use an enzyme called reverse transcriptase, which is common in viruses, which precisely copies that mRNA molecule and turns this molecule into a hybrid, an RNA-DNA hybrid. And that's what's going to happen next. So the reverse transcriptase comes in, and now what we do, the enzyme assembles all the bases and precisely copies this RNA single strand and makes a double strand of RNA and DNA. All the messenger RNA is processed in this way, but none of the ribosomal is processed. The next step is we heat the sample up, and we denature this hybrid and heat it enough that the RNA dissociates, because it's a fairly fragile molecule. So this is just chunks of RNA, and it leaves precise copies of the RNA library, which are now single-stranded cDNA molecules. The next step is we come in with another primer that goes on the other end of the cDNA copy, and this is called DNA polymerase. It now makes another copy of. Okay, again, I blew it. Let me start it up again. Sorry about that. Yeah, this will be the last time I have to do it. So we start with single-stranded messenger RNA. We make an RNA-DNA hybrid out of it, heat it up, denature it, come down and make double-stranded cDNA with a specific primer sequence on the end of it. And this primer sequence is key for the third enzyme that comes in. And we'll get there. So now we're making the first strand, which is the first complementary DNA strand with the verse transcriptase. Then we come back the other way with the Taq polymerase, which makes the double-stranded DNA with our primer A sequence on the end of it, which is coming now, using a random primer, as it turns out, here to bind to this cDNA sequence. So now we come back the other way. Now we have double-stranded DNA with a primer A sequence on the end. This primer A sequence is a promoter sequence for a third enzyme, which is called T7 polymerase. This is a common bacterial enzyme which has the property of making RNA out of double-stranded DNA, and multiple T7 polymerases can operate at the same time. We end up copying this on the order of several thousand-fold. So we've taken our original RNA library, messenger RNA library, and now we've amplified it about 2,000-fold. All the sequences in this using these three enzymes. We can do this procedure twice, and we can end up with about 1,000,000-fold. So we started with about 100 to 500 molecules on the low end. We now have 500 million molecules. And on the high end, a couple billion. That's enough to measure. So how do we go about measuring it? Again, we start with this mixture. We clean it up. We amplify it. And it turns out in one of the amplification steps, we can add bases that are fluorescently labeled. So we can actually fluorescently label each one of these molecules at the same time as we amplify it. We then use a procedure that involves what's called a DNA microarray. A DNA microarray is fabricated on a standard glass slide, and it consists of different chemicals that are deposited on the slide that are complementary sequences to the various genes that you're interested in probing. So each spatial location will selectively hybridize to a different nucleic acid sequence within this population over here, as indicated here. So basically, you spatially sort this library now. So each of these individual locations is going to have a total fluorescence associated with it, which is indicative of the original population that was in the mixture. And this is how we go about then measuring what was in that original mixture. And here's a screenshot of an actual cDNA array, which consists of about 600 spots. Each spot corresponds to a separate gene, and the intensity of the spot corresponds to the concentration in the original sample. This is part of a 22,000 spot array, which is a cancer chip that we've designed at Arcturus, which is manufactured for us by Agilent. So now we can measure 22,000 genes simultaneously in sample sizes down to about 10 cells. So this is how we go about measuring what's going on within the cell, within these small groups of cells. This is a very precise quantitative method for doing this type of analytical chemistry. Here, for example, are two separate chips that we've run, two separate cDNA arrays, and this is the intensity correlation between the 22,000 genes that we've measured. And you can see the R-squared is I believe, about 0.995 on this particular plot. So it's extremely reproducible. So it is a very, very quantitative way of doing precise analytical chemistry on biological samples. One of the questions I asked was, how do we know that cells are identical? And this is the answer to that one of the scientific questions. These are two separate captures of invasive cancer that were separately amplified and then tested on the chips. And you can see that the correlation between these two separate populations of cells is excellent. So these cells express the same genes, basically, and appear to be clones of one another, clonal populations. So it seems as though we can identify visually basically cell quanta and use that assumption is a pretty good one. If we no, those are from the same patient, but I'll show you from different patients. It also holds, that was the second question, does it go across different patients? And the answer is yes, with some biological diversity, but I'll show you that data in just a second. These are two separate populations of cells, and you can see the correlation isn't there. This is down expression, and this is up expression. Again, notice it's a log scale. So some of these genes are really overexpressed considerably from one cell population to the other. And that's what the differential expression looks like on the cDNA array. So this technology really works and works extremely well. This is what it takes to do what I just described. It takes a bunch of reagent kits and vials full of enzymes and pretty extensive manuals to show you how to go through all the steps in the amplification process. And these are what the devices look like, these injection molded devices for laser capture, for digestion, and for purification of nucleic acids. So again, the process that we're going to go through now, I'm going to show you some experimental data that we took on studying progression, genetic progression of cancer. You start with a standard biopsy sample. You microdissect the cells of interest, ending up with just the cancer cells. You then go through the process, amplify, put it on a DNA array, and then do bioinformatic analysis of the results. And you look at the expression levels of the various genes. I'm going to be showing you a bunch of data, so I want to just do a little bit about how we do the data analysis. We take a series of genes. This is genes. Here are the patients. The colors are red is up expressed, green is down expressed. This is a very important gene in breast cancer. It's the estrogen receptor gene, and it's very commonly upregulated in breast cancer. So we take, for example, all the green squares and put them over on the left here and sort all the patients that way. And then we would move all the genes that were green that went with it up to the top. So you're going to get a block of red and block of green that indicate the genes that are traveling together or clustering. And we can do this manually or we can do it automatically using cluster analysis algorithms. So this is the type of data analysis that we're doing. This is the spectra of these particular cells. So this is unclustered, so this just looks like random noise. And then when we do unsupervised learning and start to cluster, you see you get these hot spots of red and green that are associated with genes that seem to be active or inactive in these groups of patients. And this is the sort of signatures that we're looking for. In order to do this automatically, what you do is you basically define the gene expression value as an n-dimensional vector for each patient. And then you define a merit function. One merit function would just be the n-dimensional distance between the vector endpoints. Another one is the correlation coefficient, and that's what I've been showing you. That's effectively the slope of these n-dimensional vectors. We then cluster the patients by basically looking for similarities in the merit functions. And then we look to see which genes are the primary genes that give rise to this similarity. We can do this automatically, again, by various algorithms that have been developed by bioinformaticists. So these are examples. This is a typical experiment we do in this patient, which is case 43. We'll do 3 independent microdissections of the different cancers if there's more than one stage of cancer in the patient. And then what we'll do is we'll end up with a huge mass of data, millions of data points that look like this. For each patient, there's about 500,000 data points. And what we've done is consolidated this. It's about 750 genes that characterize the progression from normal cells to cancer. And these 750 genes, sets of them, this is the normal group right here, go from off to on. Others go from on to off. This is the first time this has ever been done. The technology to do this has only been around for about 2 or 3 years. And so this is the sort of experimental biology that's going on at this point within my field. You can see that for each stage, there are different sets of genes that seem to be dominant. And now we can begin to understand what the molecular biology is going on in this type of progression in disease. So if we now have an unknown sample, for example, that doesn't have the morphology to identify in any of these stages, we can just do a signature analysis, compare it with the database, and effectively do this on a very small population of cells where we may not even have to take a biopsy. So this really allows us to do essentially a virtual biopsy and obtain a cancer diagnosis without actually going through any surgery. There are other types of signatures we can look for. For example, there's not only the stage of cancer, which I've been showing you, but there are different grades. So for example, for ductal carcinoma in situ, which is early cancer, there's grade 1 and grade 3. Grade 1, you just remove the lump. Grade 3 would be probably chemotherapy and radiation as well very clear signatures that define the difference between these two types of tissue. Same thing for invasive cancer. And we can also look for signatures that define which patients would be suitable for which drugs. So, for example, this is a HER2 protein signature for the drug that's made by Genentech called Herceptin. And so we can clearly identify patient populations that should get this drug and those that shouldn't. This is an estrogen receptor signature, which is very dominant, and this is for a drug called tamoxifen. This sets of patients should not get it. These patients should get it. This is what's called the endothelial growth factor receptor, which is associated with accelerated growth of blood vessels, EGFR, and this is for a drug called Urissa. So we can also, from just a small sample of about 100 cells, tell what drug would be effective for these patient populations. Moreover, we can actually then look within these patients. These are the ER, the estrogen receptor minus ER positive groups. Here are subgroups, and we don't know what these mean. These are obviously groups of patients that have similar gene signatures, and we know, of course, what these genes are and what the proteins are, and we think they are important, but we're just in the early stages. So I kind of view this as the first time we've been able to look at a Rydberg series. So now we're developing the biology models to come up with the disease pathogenesis that would allow us to then develop more effective therapeutics and diagnostics. So the microgenomics technology that I've been discussing, really what we're trying to do is develop the technology to detect cancer at the earliest possible times by working on very small populations of cells. From the signatures that I've showed you, determine what stage the cancer is in. Also be able to tell which patients by their signatures will respond to drug therapies, which will not. And then provide a sensitive, very minimally invasive way to monitor for drug response and disease recurrence. So where we're at right now scientifically is we're just beginning now to collect microgenomic gene expression data, not only in cancer, but all sorts of other types of diseases as well. This gene expression data allows us to look for signatures of diseases, and then these signatures are going to allow us to develop molecular models for the disease pathogenesis, as I've just said, and that leads to new pharmaceuticals and diagnostic tools. There are lots of applications in the life science beyond cancer for this technology. All of these require precise molecular analysis of very small populations of cells. Developmental biology, looking at stem cell differentiation. How does an embryo grow and begin to differentiate itself and its in the different cells within the embryo into the different organs? They turn on and turn off various switches that turn on gene expression and off gene expression. And being able to measure that globally, this will be the first time that that can be done. In neuroscience, there are a lot of interest in determining what's the difference between neurons that seem to be susceptible to Parkinson's and Alzheimer's degeneration. Some are, some are not. And what people are interested in doing is measuring the gene expression differences between those sets of neurons. Diabetes, looking at insulin-producing cells and determining why some of them are susceptible to immune system attack. And in immunology, looking at basically the basic evolution of the immune response to infectious diseases. So what are the analogies to atomic physics? I think the elements of the periodic table, as I said, are the cells, spectra of the elements, gene expression data. Charlotte-Morris table, boy, we could use this, is the gene expression data. And I think the modeling that Ugo Fano and his students have done so successfully, where they use tabulated data to develop generic models for what's going on at atomic physics level, that's kind of where we're at right now, building the database so we can build these molecular models of disease. And you can extend the analogy as you go to multi-element systems. Solids and liquids are similar to tissues and organs, and you expect there's going to be all sorts of collective effects associated with building these more complex systems out of the fundamental quanta of the cells. I wanted to close. I got scooped by Mitio. This is Sunday, would have been Fano's birthday. He actually told me this back in 1978 because he was filling out a letter of recommendation for me, and he noticed that we had the same birthday, and we were separated by 40 years. So with that, I'll close.